My name is Andrea Murchak, and I'm an associate professor at Indiana University School of Medicine, and I've been instructed to go fast. So buckle up, and we're going to try and keep this on track and moving through as quickly as possible. But I think that my talk is well situated between the previous one about the microbiome and the next one, which Malutanzi will give about the immune system, because I'm really trying to understand the interplay of the communication between the microbiota and the immune system, and the bidirectional communication that we see in neurodegenerative disorders. So this is going to be taking a little bit of a sidestep. There's a lot of evidence about short-chain fatty acids, but kind of the new family of molecules in town are these bile acids and gut-modified bile acids and how they're influencing health and disease. But I actually came at this from a little bit of a different perspective. So I'm going to give you a brief overview, kind of very high level. I'm not going to be able to get into the nitty-gritty today because of time. But bile acids are generally produced in the liver. They are a part of the cholesterol synthesis pathway. They go through the bile duct, end up kind of dumped in the small intestine where they aid with digestion. There, they actually can be modified by many different types of bacteria into secondary bile acids, conjugated bile acids, changing their chemical structure slightly. These are then absorbed back into the vascular system. They go into circulation where they can kind of feed back on the bile acid production process, and the cycle starts again. And we've always thought of them traditionally in this digestive function, but there's a lot of accumulating evidence that they are actually playing a role in systems outside of the digestive system. And so this is a little bit more of a detailed illustration of the biochemical pathways that lead to bile acid synthesis. But what I want to kind of point out here especially are these different classes of bile acids: the primary, secondary, and the conjugated bile acids. The primary ones are generally the ones that are produced by our body and are being dumped into the small intestines. And then we have bacteria that can have these enzymes called bile salt hydrolases that can then shift them into these secondary bile acids. And these secondary bile acids are actually provide them a competitive advantage. They are toxic to a lot of other microbial species, can change the population level dynamics that we see of the gut microbiota. And the conjugation and deconjugation just adds these other little pieces to them, and both can be carried out by microbes. The bile acid transporters can return bile acids back into circulation, as I said earlier, but it can also be done passively, and they negatively regulate further bile acid synthesis. And I mentioned that most of the data that's been collected so far has been in the digestive tract, but we are just starting to actually look at bile acid receptors in other tissues of the body. And I, in particular, am interested in the immune system. And so here are two different bile acid receptors, TGR5 and FXR. And what I hope that you can appreciate is that TGR5 especially is present in bone marrow and spleen, these immune cells. Then we can look at the brain itself, and this is just from this review from 2020, looking at bile acid receptors. And this is just a few that I've put up here, but what I hope that you can appreciate is that they are also localized to neurons, astrocytes, and microglia. And so they are biologically relevant in the brain as well. Now, how did I fall into bile acid receptors? Why are we interested in their role in neurodegeneration? Well, I'm actually a multiple sclerosis scientist first. And so I'm going to go through my data that I collected looking at multiple sclerosis and mouse models of multiple sclerosis, and then tell you all why you should be interested about bile acid metabolism in Parkinson's as well. So for those of you who don't know, MS is characterized by autoimmune attacks of the central nervous system, where the myelin sheath that is protecting the axon is actually being degraded. And a lot of this is carried out by T cells, and specifically Th17 cells. There are about 200,000 new cases a year. It primarily affects women between the ages of 20 and 50 is usually when diagnosis happens. And there are treatments available, but no cure or preventative agents, which is very similar to kind of the state that we're in with Parkinson's right now. But in the course of trying to understand how the gut microbiome was influencing multiple sclerosis, I created these mice that have, they're missing the aryl hydrocarbon receptor, which is a receptor that we generally think of as a response element to the gut microbiota. But in the process of creating these mice, I actually found out that they have completely different metabolomic profiles. So this is metabolomic data from the cecum of these mice. And similar to the plots that you saw earlier with the microbiome data, we can see that these the individuals are very separated on this plot, indicating that they have very different metabolomic profiles. And when we look at the different, oh, this is very blurry, I'm sorry, synthesis pathways that are actually being changed in the gut contents of these mice, bile acids and bile acid synthesis is one of the primary things that's changed. And so this is too blurry to be able to see, I guess, from your side, but I'm going to point out this pink box right here, which is full of bile acids, and it's a whole network of this bile acid synthesis, with taurocolic acid being the one that is most upregulated in these mice. And so essentially what I've done is I've created these mice that have a large pool of bile acids, and it's a large diverse pool of bile acids. And we can then go through and look at their disease incidence and disease severity. And so for this, we used a model of multiple sclerosis called experimental autoimmune encephalomyelitis. And in this model, we essentially vaccinate the animals against their own myelin peptide. So they mount an immune attack against their own protein, ultimately leading to neuron damage. And when we're looking at the clinical stores here, a 0 is a perfectly healthy mouse. A mouse that's a score of 1 has a limp tail, and they're going to get this ascending paralysis up to a score of 4, which is paralysis from the waist down. And so what I hope that you can appreciate is that those mice that have high bile acids actually are able to recover from this autoimmune and neuronal damage. So I tried to simplify it a little bit by just administering taurocolic acid to these mice. So taking out that huge change in bile acid pool and narrowing it down to a single bile acid. And what you can see is that the taurocolic acid is still protective in this model, but it's a little bit of a different shape in disease characteristics. And so we needed to go through and try and figure out why that is. And so these are microscopic images of the lesions in the spinal cord itself. And what we're looking at in green are T cells, which I mentioned are the immune cells that are actually entering into the spinal cord. And in red, we have tunnel, which is a marker for cellular death. And what we found was that the T cells that were getting into the central nervous system were dying prematurely when they were exposed to bile acids. We took another measure of this. Instead of doing imaging, we did flow cytometry. And what we saw was that of these different types of T cells, we did see a higher percentage of dead cells that were CD4 positive helper cells. And so kind of what we gathered from this is that these bile acids are leading to T cells with shortened immune health spans, which also means that because they're able to undergo their normal functions, we are not, if we can manipulate the system, we won't necessarily be leading to immune compromising therapies that we have traditionally thought of with multiple sclerosis, but potentially another method. So we took this into a dish, and we were looking at apoptosis of Th17 cells, so cell death after activation in these from these same mice. And we see that those that were exposed to high levels of bile acids do die more quickly. However, if you co-house the mice, and mice do eat each other's feces, so they do share microbiomes if they are in the same housing, this phenotype completely goes away, which means that this is something that is linked to the gut microbiota and the gut microenvironment. Further, if you take the fecal contents and give them to a completely naive mouse, just basically transferring those fecal contents, we can recapitulate the cell death that we see. So we really believe that this bile acid milieu is leading to the shortened lifespan of these T cells. And then, of course, we took more of a simplified approach, and we just put taurocolic acid on T cells, and we saw the same thing. So overall, about halfway through my time, so we're going to keep moving. What I've shown you so far is that bile acids are protective against experimental autoimmune encephalomyelitis, and they lead to early cell death without compromising the ability of T cells to do their normal functions. They do not affect antigen presentation, T cell differentiation, cytokine release, or T cell homing, which are data that I did not show you because I did not have time, but are important in the context of this study. But why do we care when we're talking about patients with Parkinson's? Patients with Parkinson's have elevated serum bile acid levels. And this was a study where they actually looked at thinking about the heterogeneity of Parkinson's patients, patients that either had cognitive impairment or those that did not. And when looking at the serum levels of bile acids, these are 3 different bile acids here, you can see that across the board, those with cognitive impairment have lower levels of bile acids in their serum. Now, I'm going to go into mouse models real quick. And this was a nigral alpha-synuclein AAV injection, so this caused overexpression of alpha-synuclein in the brain. Okay, so when thinking brain first, body first, this is for sure a brain first model of Parkinson's. And again, these are plots of the conjugated different types of bile acids. So we've got taro-conjugated bile acids here in the bottom right, glycoconjugated bile acids, and what I hope that you can appreciate is those that have gotten the alpha-synuclein injections into the brain have completely different fecal profiles of their bile acids. And so even from this brain first origin of synucleinopathy, we are seeing changes in the gut. And then this is quantified, a lot of them here, and we see significant differences across the board between those mice who have had this injection of alpha-synuclein AAV and those that have not. And so this is a quick study that was done in rats with an MPTP model, looking at how this taro-ursodeoxycholic acid can be protective in the MPTP model. And so the design of this was we had certain mice that were vehicle, no treatment whatsoever, the MPTP, so we should see degradation of the neurons and behavioral deficits. And then those that were given the bile acid either for 3 days prior to MPTP administration or 3 days after. So kind of a prophylactic and a treatment paradigm. And those are shown here in these 2 bars. And what they saw was differences in the number of Th positive fibers that were remaining after this administration, meaning that there was some protection by bile acids against the MPTP model. They went through and did some behavioral analyses and were able to show that this was also protective on the behavioral front. And then when they looked at neuroinflammation, looking at GFAP and at IBL1, respectively, they also had protective effects in reducing the inflammatory state of these, the brains of these mice. And so I'm going to leave you with, I've talked a lot about the immune system and the adaptive immune system and what bile acids could possibly be doing there. But ultimately, I'm going to be leaving you with more questions than answers, unfortunately, because we don't know how it's affecting the immune system. There's been a series of work going on for the last several years leading to this hypothesis of how one particular receptor called TGR5 is influencing neurons, microglia, and astrocytes themselves, but how those are then interacting with the immune system, both the adaptive and innate, we don't yet know. So big questions that I'm trying to answer with my career and the projects that I'm working on with my team are why and how are T cells being modified by bile acids? Which receptor is causing the changes? Where are they seeing the bile acids? Is it in the tissue? Is it in circulation? How can we effectively target neurodegeneration while avoiding off-target effects? Bile acid metabolism is very complicated, and it's really important for cholesterol homeostasis. It's important for digestion, all of these features. And so we need to make sure that we're not disrupting other things that, are already causing difficulties in patients with Parkinson's. How can bile acid metabolism be harnessed to promote health? So is this going to be a drug target? Are we going to be repurposing drugs? Is it going to be a dietary intervention? And, potentially, can probiotics fulfill a clinical role for bile acid metabolism? So one thing that we're working on right now is we have these bioengineered bacteria that overexpress these bile salt hydrolases and hopefully can kind of pick up where our endogenous bacteria are not able to keep up with the needs that we have. And so with that, I'd like to thank the team who has helped with a lot of this work and is going to be working on it as we continue. But I think I've done pretty good on time, and so I'll take any questions.
I have a question. So you're looking at wanting to increase the bile salt hydrolases. Mm-hmm. I'm going to say maybe a possible issue with mm-hmm. most of the commercial probiotics. Mm-hmm. They are actually selected. So you select strains of bacteria for probiotics in the market mm-hmm. to be bile salt hydrolase resistant. Mm-hmm. So certain bacteria then sort of in the food supply could be passing on that resistance.
Oh, transferring the resistance, yeah, absolutely. The resistance. Yeah, absolutely. So, yeah, that is the chronic problem with using probiotics, right? Is that they are not stable. Different people are able to be colonized to different degrees, and how much is actually getting into the gut and remaining alive is kind of a question. And so, ultimately, is it a question of the actual bile acid metabolism itself, or is it a question of the probiotics that are living and the bacteria that are living? And I think that we've not fully teased that apart. When we tease that apart, then we can more easily target which ones we want, I'm going to say. Yeah. Okay.
We should probably go on to Malone, and then we'll cover more questions.
Sounds great.
So we're getting closer.